Hello everyone. I would like to start by thanking the organizers of the meeting for this amazing opportunity to talk about my work and also the opportunity to visit Korea. It's been an amazing experience for me. My name is Bárbara Fonseca. I work at the Biomaterials and Microfluidics facility, and my talk will be about stem cells and how to use these cells to develop better models of disease. So we all know that during the drug discovery pipeline, or in general for advancing biomedical research, it's necessary to use different types of model systems. And we know that usually in the vivo side, we have different animal models that are really highly relevant for many steps, but sometimes can lack some due to differences of human specific pathogens or human-specific traits. Sometimes animal models cannot completely recapitulate all the complexity of the human-specific biology. On the other side, in vitro systems, such as the use of immortalized cell lines, are really useful, especially for the initial steps of pre-screening, high-throughput screening, molecular biology studies. But because of their simplicity, at the same time, they can not fully recapitulate the complexity of the human tissues. So that's why many regulatory agencies in Europe, in US, and also here in Korea are promoting new approach methodologies to try to deliver human-relevant models that can address complex diseases and then foster innovation. And among them are generally what we call microphysiological models that are attracting a lot of interest, and with the perspective to maybe build more predictive models. So among them, I can give a small definition of both that are mostly used, that are basically organoids and organ-on-chip. So basically, organoids are considered as a simplified version of an organ produced in vitro. It's not exactly a mini organ, but it's a way of culturing the cells, usually in 3 dimensions, that because of this type of culture, they can mimic some aspects of an organ. It's basically composed of stem cells, and because the stem cells can also give rise to different cell types, they can self-organize in vitro, and then form some structural and functional aspects of an organ. So I can see, for example, hormone production for many different types of organs, such as placenta, thyroid, pancreas. You depend on this 3D organization. In the case of the brain, to have the spatial regionalization of the neuronal layers of the cortex, you also depend on this 3D organization and so on. So these are basically the organoid culture system. On the other side, we have the organ-on-chip. That's, as a definition, is quite the opposite of organoids. So while in the organoids, we are counting of the ability of the stem cells to self-organize themselves, for the organ-on-chip, because it's an interface between biology and engineering, we try to mimic try to control, in fact, the microenvironment that the cells are. So we see the cells in a way to try to control this microenvironment, to try to replicate some physiological functions, such as the tissue-tissue interaction, 3D topology, biochemical microenvironment, as I said, and also the mechanical forces. So in these images, there are some examples of an organ-on-chip, but I will give one more precise one. That's the simulate technology that was already introduced by UNMed before. This is one of the first organ-on-chip device established, and is still one of the most used in the field. It's basically a small PDMS device that you can culture cells in 2 different chambers. So they are separated, but at the same time, there is some level of communication because they are separated by a membrane with a pore, and this pore allows not only communication of media nutrients and so on, crossing of the microorganisms if you want to mimic epithelial crossing of a microorganism, for example, and also recruitment of immune cells. So this can be allowed by this type of device. At the same time, you have a microfluidic control, and as you can see also in the video, you can mechanically stretch like in a cycle way to try, for example, to mimic, in the case of the lung, the breathing movements that happens during respiration and that the alveolar cells sense. And also, in the case of the gut, the peristaltic movements. In that sense, many years ago, our lab used this type of organ-on-chip device to ask the question if the 3D microarchitecture and mechanical forces in a gut-on-chip, for example, could impact Shigella infection in the epithelial barrier. And this, so for that, they did this human gut-on-chip device where you could mimic this typical topology of villi and crypt of the gut, the human gut, and they could see that Shigella infection not was only more present, but was stronger in the intestine chip. I'm going to try, yes, here. Intestine chip in compared with a conventional 2D system, I'm sorry, showing that the, using this type of chip is really interesting to mimic this type of infection. So we just stop now just to tell a bit about us. So we are a core facility at Institut Pasteur that part of our work is based on training different uses of the institute in all these types of microfluidics engineering applications, also for organ-on-chip and more recently in organoid technologies, so they can train and autonomously use these models. But at the same time, because this technology is constantly evolving, we are also involved in different co-development projects. So we also are co-developing different types of model systems. So here I'm just listing a few ones. So as Guillaume already showed, we together with Elisa Chakrabarti, the lab developed the lymph node organ-on-chip model that's being now currently used to test some vaccines and etc. And we also, in the case of organ-on-chip, we have gut, nose, airway, alveoli. And in the case of organoids, we also doing gut, lung, and more recently placenta and brain. So, but today I will focus more in the personal project that I've been working more in the last years. That's the lung organoid on chip to model infectious disease. So here, the objective is try to combine these both approaches. So we use organoids as a source material to seed our chip to try to create these models. So here, just a quick overview of the respiratory tract. So basically, as you may know, the respiratory tract is the first barrier against respiratory infections. So it has a barrier function, and it secretes mucus and surfactant, and has a strong host immune response. And at the epithelial level is basically separated by two types of epithelium. That one is the conducting airway epithelium, mainly formed by secretory cells and ciliated cells. Then together have the mucociliary clearancing capacity to sweep out the system all the pollutants and toxins and microorganisms that try to invade the system. And in the most distal part, in the lung per se, we have the alveolar epithelium, that where the gas exchange happens, and is mainly composed by type 2 pneumocyte, or alveolar type 2 cells, and type 1 pneumocyte, or alveolar type 1 cells. So this is the general knowledge that we know about the respiratory tract. But interestingly, in recent years, because we have an advent of transcriptomics, spatial transcriptomics, and also single-cell transcriptomics, we know that in the distal region of primates, so human and other primates, there is a specific region between the airways and the alveoli that's called this transition zone, okay, here, that we have cells that express a mixture of key markers of both airway and alveoli. And we don't know yet, it's not completely understood what is the specific function of the cells harboring these two lineage markers, but it's been described that these cells may be involved in some pulmonary disease, such as idiopathic pulmonary disease and also COPD. And also, as the entire respiratory tract, this region is also invaded by pathogens, such as pneumococcus, RSV, SARS-CoV-2, and so on. So we decided to create such a model system. So our strategy was to use the human feet on lungs, that we can isolate the cells, to do a primary culture and culture them as organoids. And because we can use a specific media composition to maintain them in a multipotent self-renewing state, we can amplify the initial material and bank. So we have already more than 30 donors already in our liquid nitrogen, and they are maintaining this multipotent state. And when we decide to do the experiment, what we can do, we then thaw them, we put them in culture, and then we can trigger the differentiation. And we can trigger the differentiation towards distal lung organoid or organ chip. And which method we're going to use is going to depend on the downstream analysis. So for example, for infection studies, I mean, lung organ chip is really interesting because you can put the cells in a way that you can expose the apical part of the epithelial cells that are usually where is the receptor of the pathogen is. So that's it's an easier way to perform infection studies. So, yes, so here is just a few images of this distal lung 3D model. So on here in the left, you have these organoids with key markers of alveolar cells, more specifically alveolar type 2 cells. And we can see this, find the same markers in this lung on chip. So here are images of a top view of cells on the chip. And what is interesting is that these cells, they form an epithelium tight because we can see the expression of ZOO1. And also, this is a particular thing that result that we see in the organ chip, that we see the appearance of alveolar type 1 cells that we don't have in the organoid system. So if you are interested to understand the biology of the alveolar type 1 cell specifically, the organ chip is a good strategy. And recently we did a single-cell RNA-seq profiling of these distal lung models. So basically what we saw is that this SFTPB, that's the key marker of this distal lung lineage that I told you, that's like this transition zone plus alveoli, it steadily increased during the culture system and achieving mostly 90% of the cells of the chip that express this marker, showing that they are, at least this key marker of distal lung is present. And we look carefully over all the cell populations, we see that we have an increase of alveolar cells, typical AT1, AT2, AT0, so it's around 10%. And we have the appearance of this, it's not working, yeah. This appearance of this terminal respiratory bronchiolar secretory cells, that they are about 40% of the cells on the chip. And they are, and together with the alveolar type 0, they are considered these cells that are more involved in acute lung diseases, such as IPF and pneumonia, as I mentioned before. With such a system, we model Streptococcus pneumonia infection. And just a quick note, pneumococcus is considered kind of a commensal bacteria when it's residing in the upper respiratory tract, but colonization in the lower respiratory tract can cause pneumonia and can be severe depending on the serotype of the bacteria. And we could, with this chip, mimic the infection with this bacteria, and also in the video you can see the crossing. You can see some bacteria that were identified in the bottom channel, so just they crossed towards below the epithelial barrier. And we've been working for a while with SARS-CoV-2, and for a long time we see that we can infect these chips with SARS-CoV-2, so we can see in red the spike-positive cells, and in the yellow arrows, cells that are co-expressing an alveolar marker, H2-280 in spike. And what was interesting is that when we compare the effect of different SARS-CoV-2 variants, so Delta versus Omicron, we can see a clear difference of replication capacity in these chips between these two variants. And this result we could recently confirm by single-cell RNA-seq. So we identified cells infected with the Delta variant, but not with the Omicron. And also we saw that the infection with Delta can induce a strong interferon-stimulated gene signature in the cells, in the bystander cells, not exactly in the cells infected, but the cells that are around. And in conclusion, this data corroborates what we see in the literature in vivo, that the Delta, but not Omicron BA.5, can replicate well in the alveolar lung chip, in the distal part of the lung, and supporting this in vivo and other in vitro evidence. So this was a really nice result. Just mention briefly that we are continuing this work, so now looking more specifically in the mechanism of the host response using these models for SARS-CoV-2, pneumococcus, and recently with TB in an internal collaboration in Institut Pasteur. We also are working with an immunocompetent lung-on-chip model. This is the work of a new postdoc in the lab. Because I didn't mention, but for the moment we have basically an epithelial compartment, but the idea is to create an entire complex chip with a vascular compartment and adding immune cells. So more cells we add, more complex the model system is, so it takes a bit of time of performing this. But Jake made a really nice, he has a really nice results, so you can see here in the image the vessel formation in the alveolar lung chip model. And also he's working in a different custom design chip for other applications to do like perfusible vasculature. And finally, regarding lung-on-chip in the lab, this is the work of a PhD student, Sophie, that in the collaboration with Michael Connor from Elina Mon's lab. And they are designing a specific custom-made chip design where they are trying to replicate in vitro the geometry of the nasal compartment in a chip model to try to replicate the nasal airflows on chip. Because the idea is to study exactly how the bacteria, when you have these breathing movements, how they're going to colonize specific niches inside the nasal epithelia. So basically, this is the work she's doing. It's really nice. And finally, just to mention me personally, I'm working now with brain and placenta organoid models, so we are establishing different collaborations. And for these both models, we are using iPS-derived organoids. And if you want to discuss further, please contact me. It would be nice to talk. And I would like to finish by thank you all the facility, all my colleagues. If you are interested for the organ-on-chip technology, I advise you to discuss with my colleague that's called Min-hee Kim. She's a Korean researcher. She's a really talented engineer, so she can help you as well with the organ-on-chip technology. And I would like to thank the collaborators, the funding, and you for your attention. And that is it. Thank you.